Okay, so in this video we are going to talk about the formation of an adaptive T cell response against some form of generic pathogen. The whole process takes a couple of days, up to a week, and starts with the innate immune system. The innate immune system is actually critical for the formation of an adaptive response, or in this case, T cell activation, and without it, you're pretty much screwed. Okay, so in this case, I have drawn a pathogen here. This is a bacteria, but it can be pretty much anything like a virus, protozoa, fungi, or whatever. Um, and this pathogen has somehow been able to cross a bodily barrier. It can be the skin, the lungs, the guts. It's basically on the other side now through an injury or something. And the first thing this pathogen is going to encounter is a bunch of stromal cells, or basically cells that make up the actual barrier. And they are maybe not directly immune cells, but they are for sure part of the innate immune system. And they can recognize pathogens through a type of receptors called pattern recognition receptors, or PRRs. Um, and they kind of are expressed all over the place, both on the actual outside, the plasma membrane, and also inside the cells, both on like vesicle membranes or in the actual cytosol. But these pattern recognition receptors are able to recognize both structures from the pathogen, such as pathogen associated molecular patterns or PAMPs, as well as danger associated molecular patterns or DAMPs. And these PAMPs can be many different things, like parts of the flagella, parts of the membrane of the bacteria, and also DNA and stuff. And the DAMPs can be basically stuff that isn't supposed to be on the outside of the cell, so ATP and DNA. That kind of signals that there's something bad going on and that we need a reaction here, we need an immune reaction. So basically all of these binds to, or can bind to, different pattern recognition receptors on the stromal cells here, which will kind of lead to them altering their protein expression pattern. So now they will be able to secrete cytokines. Especially, or maybe most commonly, IL-1 beta, and also TNF alpha, just to mention two of them. And these cytokines will be important later, and I will come back to them, but there's another cell type here in the vicinity that's crucial for a T cell response, and that's the dendritic cell. Let me just draw it here. So this is a dendritic cell, and as you can see, it has a bunch of dendrites, which it uses to kind of scout and probe the microenvironment for stuff that's not supposed to be there. Yeah, that kind of looks like a neuron without an axon, and these are found in various tissues, and they are crucial for a T cell response. Without them, we wouldn't really have an effective T cell response at all. And exactly as like the stromal cells, these cells also express pattern recognition receptors. And two of the most, I would say, important one in this case is the TLR family. Stands for toll-like receptors, and they bind to, depending on, we have many different ones, 1, 2, 3, 4, 5, 6, 7, 8, 9, and they are expressed at different places at the cell. Different cell types expresses different combinations of TLRs, but here we're talking about, it could be perhaps TLR 4 or 5. They basically signal through the same pathway, so it doesn't really matter in this case, but they are able to recognize conserved structures on pathogens. And in this case, let's say it's the flagella of the bacteria, then it would be TLR5. If it's LPS, it could be TLR4. But when our pathogen here is in the vicinity, products from it, precisely like in the case from the fibroblasts and the epithelial cells, products from the bacteria is able to bind to these TLRs to trigger a pathway called the NF. Let me just NF kappa B pathway. And this pathway is pretty much one of the most important pathways in immunology, I would say. Then the dendritic cell also uses another type of pattern recognition receptors called CLRs. Which stands for C-type lectin receptor, and CLR binds to conserved carbohydrates on the bacteria, so different kind of sugars. And binding to CLRs triggers phagocytosis of the bacteria into a phagosome. So there's the bacteria, and it will be inside the dendritic cell. But CLRs also can lead to NF-kappa B activation. And if you remember the cytokines from earlier, these will also play a role in the context of dendritic cell activation, because these cells express various cytokine receptors, and especially TNF-alpha as well as IL-1 beta is really good at activating dendritic cells. So fibroblasts and basically the stroma, together with the actual pattern recognition receptors on the dendritic cell, will help activate it. So now we have an activated dendritic cell, and what's going to happen now is that it's going to start to migrate to a regional, or basically a close lymph node. And this process is mediated through the upregulation of a couple of, or especially one type of chemokine receptors, and that one is called just CCR7. So the dendritic cell will upregulate CCR7, which is the receptor for its ligand CCL21. And CCL21 is expressed by stromal cells inside the lymph nodes, the secondary lymphoid organs. And this will create a concentration gradient which kind of attracts the dendritic cell, because they follow the gradient. And they will enter the lymphatics and move towards higher concentrations of CCL21. And together with CCL21 and all the receptors we talked about before, it will also start to upregulate MHC molecules on its surface, both 1 and 2. And MHC molecules, they function by presenting peptides, both self-peptides and microbial peptides. Class 2 only presents peptides from pathogens. And what's also going to happen is that the co-stimulatory receptor B7 is also expressed on the dendritic cell while it's traveling through the lymphatics. And this B7 expression, it's really what sets the dendritic cell apart from other types of APCs, such as macrophages. They express these to very high concentration levels in comparison to macrophages. So they are suitable for activating naive T cells. What's also going to happen is that the cells are going to stop phagocytosing stuff. So they are basically, they have eaten what they have eaten. They will not engulf anything more, and they will start to process it on its way to the lymph nodes. So I'll just put secondary lymphoid organ migration. So this process takes a couple of days, and when they arrive, they use the lymphatics to kind of end up in the T cell zone of the lymphoid organ, and that's perfect because that's where the T cells are, as you can imagine by the name. And now when they are here, they will start to secrete their own type of chemokines, and one of those are CCL18. And what CCL18 is going to do is it's going to provide as with the CCL21 before, it's going to provide a chemotactic gradient which attracts primarily naive T cells to the dendritic cell. So that's the cells that haven't really encountered antigens before is going to come to the dendritic cell. And now, thanks to CCL21 and the pattern recognition receptors being activated, it's going to express the MHC molecules, which kind of looks like this. On its surface, and these molecules, as I said before, present small peptide chains from the pathogen in the binding groove of the MHC molecule after it has been digested and then presented on here. They are also going to express B7. Let's put MHC. And B7 is also called CD80 and CD86, depending if it's B7 1 or 2, but I'm just going with B7 because I don't really like all the CD names. I know in immunology that's most of the proteins, but yeah. So now the cell is ready to present the antigens to the T cells, and it's going to encounter a bunch of them. And many of the cells aren't actually going to be able to react to the peptide because their receptor is specific towards this one. But sooner or later, one T cell will show up with a receptor that's able to recognize both the actual peptide itself as well as part of the MHC molecule. So it will bind to both the MHC molecule and the peptide. It's called a TCR, T cell receptor, and it actually consists of several different subunits. So you have the CD3, which does the signaling, and the alpha and beta, kind of looking like a B cell receptor. They are in the same family, but this interaction is not enough to activate a T cell. Let me just put. I'll name it T0 because it's naive, and we are talking in this case, this specific case here, about naive T cells, naive T helper cells. So this interaction alone is not enough. It needs B7, and the receptor for B7 is called CD28. And without this interaction, the cell will become anergic and go through apoptosis after a while. And it's actually a mechanism of tolerance. If a dendritic cell is activated or kind of travels to the lymph node without recognition of pathogens, it won't express the B7, and thus it will kind of eliminate autoreactive T cells, but that's another video. So, as I said before, TCR signaling alone is not enough to activate a naive T cell, but together, both of these can bring the activation threshold high enough. You get a signaling cascade that's strong enough that it's going to activate P13K ACT pathway as well as the MAP kinase pathway. And this will lead to a bunch of different stuff. You will get NF kappa B, you'll get AP1, as well as NFAT. And these are basically the most important ones. These transcription factors are the most important ones in T cell activation. And all of them together are needed to generate a steady expression of cytokine called IL-2. And IL-2 is really perhaps the most important factor here. Because when IL-2 is produced, it will be secreted by the cell, and it will go back and bind to its own receptor on the same cell, IL-2 receptor. Because when all of this happens, the cell also upregulates IL-2 receptor with the high affinity subunit CD25. So now it binds IL-2 very strongly, and it will secrete it to promote several different pathways, among one of them being activating the mTOR complex, and which will lead to the upregulation of various different cyclins, which, when you have cyclins, it will increase the cell cycle, so the cells will go through the cell cycle quicker, and it will also lead to an upregulation of anti-apoptotic proteins such as BCL2 and BCLX. So now basically the cell is able to proliferate quickly and not die, thanks to the IL-2, which in part comes from all of these signaling pathways. I can make a more detailed video on all these things here if that's something you feel like I should do. So that's two of the most important parts here, but there's a third factor to all of this that's also important in the activation of T cells, and that's also coming from the dendritic cell, and that's cytokines. And these cytokines will bind to various different cytokines receptors on the T cell, and they more or less determine which type of, if we are talking about the CD4 positive T cell here, it will determine the polarization program. So we'll get a Th1 cell, Th2, just 1, 2, or 17. All of these require different cytokines. So Th1, you want interferon gamma and IL-12. IL-2, you want IL-4 and 13. Th17, you want IL-6, IL-1, TGF-beta, and stuff like that. Because they induce different transcription factors, which kind of determines the polarization program of the T cell. And depending on kind of which environment the dendritic cell resided in before it moved into the lymph nodes, that will kind of determine what kind of cytokines you get during this phase here. So that's called DC conditioning, and it depends on what kind of environment it lived in before and what kind of pathogen binding to different pattern recognition receptors in the dendritic cell. And that may also be another video on its own. But this is just a generic T cell activation video, so I won't go into that further now. But now you have all of these three signals. You have cytokines, the TCR, and the co-stimulation. And now several different things is going to happen. So to begin with, these cells here are going to proliferate, as I said before, generating a bunch of different, bunch of clones of this cell here with the same TCR, so they will be able to recognize the same peptide here, and these are the effector cells, more or less, and they will just name them for short, and they will start to upregulate chemokine receptors. Upregulate chemokine, just CCRs. So depending on where the actual initial infection occurred, so depending if this happened in the lungs, the gut, the skin, that will determine what kind of chemokine receptor we get here. So for the skin it might be one, for the lungs it might be another, and some of them will actually gain chemokine receptors that doesn't correspond to the original tissue infection. So they will kind of move to another tissue and establish memory there to give global immunity. If you get the infection somewhere else, instead of the skin, you might get the same bacteria in the lungs. You want protection there later. So now the effector cells are formed. They will, as I said before, upregulate their chemokine receptors and start to migrate to the tissue where they are needed the most. So where you actually have the ongoing inflammation and infection. So they will migrate here. Sorry for that horrible arrow. And now the effector cells um, can secrete various cytokines to help the innate immune system clear up. They can provide direct mechanisms by killing infected cells, and they basically help clean up the mess by secreting mostly cytokines, but they do a bunch of different stuff, and that's also another video on its own. But some of the cells doesn't travel back to the tissue. They stay in the lymph node to become another type of helper cell called follicular helper cell, and a small fraction of them become these cells, and they will stay in the lymph nodes. They will migrate towards the B cells, where they will provide help with high affinity antibody production. And kind of helps form a robust memory B cell response as well. So we get memory B cells. You don't really get that without these cells here. And another important function of these T cells is that they will also start to upregulate a receptor called, or a ligand, more like it, for a receptor called CD40 ligand. And CD40 ligand is going to bind to CD40 on the dendritic cell, and this will lead to even higher expression of B7, making them even better at activating other naive T cells. So there's kind of a loop going on here, and all this will occur until you have a clearance of the antigen and the pathogen is removed from the body. And then most of the cells will go through apoptosis, but some of them will form a memory response, so we get the lifelong memory, hopefully, against the pathogen next time it infects you. But that's all for this video. Thank you for watching.